Hello and welcome back. Today I'm going to present my second video on point pattern analysis, where I'll begin a discussion of sampling methods that have been used for point pattern analysis, taking a somewhat historical approach to this. So we can look at some of the ways that sampling has been done. Initially, this was done using something called plotless methods or quadrat methods, which were done by random sampling. Later on, as the techniques improved and also the statistical techniques as well as the sampling techniques, we started incorporating things like survey techniques, which had been developed for surveying land and are quite good at accurately locating different positions and giving them coordinates. We'll also consider things like remote sensing, which is a more modern technique where we can sample very large areas if we have the appropriate way of finding and assigning different locations in the image to the objects that we're interested in studying. So in this first part, talking about methodology, we'll focus a lot on some of the historical approaches that were useful and used quite a bit in ecological studies and predated some of the more advanced techniques we use today, given that the statistical techniques have gotten more precise and more useful as the technology has also improved to do this kind of work. Before describing these methods, however, I'll first review the primary context behind how these earlier approaches thought about spatial point patterns. There are really three basic kinds of pattern in terms of point patterns that are considered as a first sort of step in understanding whether a distribution of points like individual plants or disturbances like gopher mounds have any spatial structure at all. So the first possibility that we consider is the random condition, so random. This is a situation where there's no spatial pattern at all, and there are actually two components to having no spatial pattern, at least in a technical sense, if we think about the mathematics or the geometry of what's going on. The first step is that individuals are independently distributed, and so this means that an individual will come into the system, and the first individual and any other individual that appears, such as the second one here, will have no influence from the first individual as to where it can occur. This occurs for all the points or objects that occur in the system. They appear wherever they land, and there's no influence of anything around them that changes where they occur. The second component of this is, and I've already implied that from the previous description, is that there are really no spatial interactions among the individuals. Now, for a spatial ecologist, this is a really boring case. This is what we call the null model. So it's a system where there's no spatial interaction, there's no spatial structure, and so for a spatial ecologist, there's really nothing we can study. Although this can be interesting if there isn't a spatial interaction, this may be interesting as well to ask why isn't there, because it seems like there should be. This situation of complete spatial randomness, where we have this process, is often referred to as CSR, or complete spatial randomness. And if you read papers about point pattern analysis, this is a term that's often used. So we have this as sort of a test case. So as opposed to that, if it's not a random process, there are two possibilities that we might encounter. The first one is perhaps the most common, and this is where we have a clustered process. So in a clustered process, they're not just distributed here, there, and everywhere, but we have areas where there are a lot of points, and then we have other areas where there aren't any. And so this clearly has a fairly strong spatial structure. The reason an ecologist is interested in this is that this can imply a couple of things. One, it can imply that there's a difference in microhabitats. And so the reason that we see a cluster here and a cluster there may be because there are different nutrients available. So these might be areas with high nitrogen, so the organisms can grow there or be there, and other places are low in nitrogen. So this can cause a pattern in the system because of this background difference in habitat. Another thing that might be going on that's of interest to ecologists is that there is a limited dispersal from an individual plant or animal for the offspring. And so if we have a new individual come in, it might come in there and then produce offspring, and they don't go very far away. And so we get a clustering process like that. And that may be another reason that we see this kind of clustered pattern in space. So when we do a study, if we determine that it's clustered, these are the two things that are often explored. Does it imply that there's a difference in the background environment that's causing this pattern, or is it because of something going on biologically, such as limited dispersal distance? So that's one of the possibilities if we have a non-random process. The next one is that we have a process that causes what's often referred to as an even distribution. But I prefer actually the term hyperdispersed because this is a more biological kind of concept. Even implies that there's some regularity in the system, and although in this example here we see a rather even distribution, if we think about it in terms of the biology, this could be caused by, in many cases, competition for resources, which is the primary reason we think this might be occurring when we see it in a natural system. And the reason for this is not because there's something in the environment that imposes that you be very evenly distributed. It's more that there's interactions that prohibit individuals from being too close to one another. So an example of this in plants would be competition in the root zone may keep other individuals from growing nearby. There can be chemicals exuded by the roots that prohibit growing near a larger plant, and so we see this producing a distribution that looks even, but it's hyperdispersion because individuals can't abut with one another. In the animal world, this can be due to things like strong territoriality. So we can see this kind of hyperdispersed pattern, say, in the distribution of ant mounds and things like this. So these are basically the three different categories we think of in terms of point pattern analysis as being produced, and what we try to do is differentiate between the three when we do an analysis. The earliest approaches really focused on this, although now we're a little more subtle in how all of this can be done. So we can shift now to a consideration of how we might sample a pattern to analyze whether or not it's a random, clustered, or hyperdispersed pattern. The techniques that were used initially were developed by foresters where they employed what are commonly known as nearest neighbor plotless methods. So these methods have taken a number of approaches where we find a system where we have points distributed throughout. If you're a forester, these are going to be trees. So this could be a forest where we have trees distributed in this fashion. And then the approach using nearest neighbor techniques was to measure a number of metrics that can give us insights into how closely packed plants are or whatever we're measuring. It can also be used to determine if they're hyperdispersed or clustered. And so there was a huge explosion in metrics that were suggested for how to do this. And so some of the techniques involved locating an individual and then finding its closest neighbor and describing that distance as the nearest neighbor distance. This is done by repeatedly finding a large number of plants, choosing them at random or mounds or whatever, and then measuring this distance W for a whole suite of instances, and then using that set of numbers to ascertain whether or not it's a random pattern. If it's not a random pattern, then we can determine if it's a hyperdispersed pattern or a clustered pattern. So that's one of the simplest techniques. Another technique that is used is to take the second nearest neighbor. So we have the nearest neighbor that's there. We find it, but then the measure that we use is the second nearest neighbor, which we can call x2. So this is a metric that was also used, and there were arguments in the literature as to which one of these gave you a better sense of the true structure of the system. Other approaches are to find the nearest neighbor from the closest randomly located spot. So we don't start by finding an individual, we just find a random point, and then we find the nearest random location to that. And we could call that something like X, or we could find the nearest point to the nearest individual and then look for the nearest individual to that, which is basically two steps with starting at a random point, and we can denote that by something like Y. There are even more complicated measures that were devised to try and ascertain what the nature of the pattern is. In one particular textbook I know, there were at least 17 metrics that were used using these kinds of measures in various combinations to try and determine what the structure of the pattern was. And there was argument among people developing these as to why theirs was the best versus the other ones, and they all had various properties. So from these test statistics, the basic idea was to develop what the CSR, or completely spatially random expectations, were for these measures, and then use those to test against the expectation as to what you really actually see in the field to come up with what kind of pattern you actually have. A second approach to this that is one that has been used more commonly by plant ecologists, at least historically, were quadrat methods. And so this is similar in some ways, but different in another aspect, in that what you do is you take a quadrat, which is shown in this frame here. It's made from PVC pipe or plastic pipe in a square, and you randomly throw it into the field, and then you count the number of individuals or points that occur within that quadrat. Now, this is only one possibility. Some quadrats aren't square, they're rectangular. Others can be circular. They can be basically any shape that is easy to construct and use in the field. And when we use this approach, what we do is we go out into the field. This may be an area here that we're interested in studying. And so what we do is the first step is to throw out quadrats in a random pattern and sample within each one. Within each one of those quadrats, what we do is we determine the number of objects that we find. So if it's plants, we determine how many individuals possibly, or if it's gopher mounds, how many gopher mounds we found in it, and use these as the data that we test against what our expectation for a completely spatially random process is. And so once we have the data assembled, we have the number of quadrats that have different numbers of individuals. So we may have 10 that have no individuals in them. We may have 5 that have 1, 7 that have 2, 10 that have 3. And so we use that to test against what we would expect for a completely random process given the average number of points that we have in that quadrat. So a typical way of doing this is use a Poisson distribution. You possibly have learned about that in the course elsewhere. So this is a typical assumption in terms of a random process for a point pattern. So we can test against what it predicts the number should be, the number of quadrats that should have each number of individuals in it, and do something like a chi-square analysis to determine whether or not it's a random process or not. So the assessment of the process is really a two-step thing. And if you've had some statistics, you'll appreciate why it's done in this fashion. So what we do is we start with our pattern. We have our data. and we test it using, say, a chi-square test or some other appropriate test. And then what we do in the first test is ask the question statistically, is this pattern random or not random? So what we do is we test it, and the only thing we can do is to show that what we've sampled is not showing the same that we would expect for a random process. And if that's the case, we can say statistically that we have a non-random process, which to us as a spatial ecologist is nice because we're interested in spatial interactions. So once we've done that step, if we can indeed demonstrate that the pattern isn't a random pattern, then we can go to the next step where what we do is differentiate between the two options for a non-random pattern. So again, just to reiterate, it can either be even or hyper-dispersed, or it can be clustered. So what you would see in an even pattern using the quadrat technique is that you would on average have the same number of events within a quadrat if the quadrat's big enough, because they're evenly dispersed and you're going to end up getting pretty much close to the mean number of individuals each time. If you have a clustered process, what you're going to find is that some of the samples have a large number of individuals and other samples will have none. And so there's a huge difference between and much higher variability and variance in what you see. And it's this that we can use to determine which of these two cases it is. In the even case, there's little variability between samples. In the clustered case, there's high variability among the samples. So we look and see whether statistically the variance is higher than we would expect for a random process. And for the even case, it would be that the variance is lower than we would expect for the random process. So again, we can do this two-step process in our analysis. We first determine whether or not it's a random process or not. If it's not a random process, then we can assess which of the two possibilities it is in terms of even or clustered as a pattern. So these are some of the traditional approaches that were used quite extensively until the advent of newer techniques. And the reason that this was done for quite a while was because of the statistical techniques that were available, and also because it's fairly simple to apply these approaches in a technical sense and terms of time it takes to do the analysis. But as we got more experience in these techniques and learned more about how a pattern is structured in different systems, we started to want to ask more sophisticated questions. And so there are a number of things that we have to take into consideration if we want to improve our ability to analyze point pattern in particular. And one of the major things that's a criticism of the quadrat methods is that they're really sensitive to size and shape. So if we have a round quadrat, we get a different answer potentially than if we have a rectangular quadrat, and the size of that quadrat is going to change how we assess the pattern in the process. For example, in terms of size, we could have really small quadrats like this, and we would have a very different sample than if we had really large quadrats like that. And so our answer may actually differ as to what kind of pattern we have depending upon the size and shape of the quadrat. So this presents a problem in that our answer may depend upon our technique in a way that we're not particularly happy with. The other thing that was a problem with the original sort of concept of how to do these analyses and the original approaches is that the metrics that we use to assess the pattern are not really well understood away from random. So although we can say it's even or clustered, we can't really be very detailed in what that means. It's just sort of three possibilities without a lot of nuance. And so that's a problem. The other thing that was something that people in the field who are interested in point pattern analysis were bothered by is that you have one answer for a system where pattern may be structured in a very complex way, and this could lead to missing out on what's really going on. For example, the pattern that we see here may have a hierarchy of different levels of pattern. And so the approaches are going to miss a lot of what's going on potentially, and would like to be able to tease apart what that pattern is actually doing. So we'd really like to take sort of an approach that may be able to tease apart what's going on at different scales. And let me just give an example of what I mean by this that can perhaps be descriptive of the way a pattern might develop. So a lot of the new techniques in pattern analysis, many of which are shown here, which you may be familiar with, such as Ripley's K or Ripley's L. We have pair correlation function here, and a couple of other metrics as well shown on this diagram. One thing that you'll note here is that the x-axis on this diagram is on the scale of meters, and this is what the scale of the process is. And so these techniques give us information about how the pattern changes across a range of scales, not just whether it's random, and if it's not random, whether it's even or clustered. We can assess this at different measurement scales and get nuanced answers as to the structure of the spatial pattern. What this shows here are the CSR expectations for the pattern, and we might see, for example, if we're looking at this measure here, L1, that when we assess the pattern with this metric, it may do something like that, and we have a pattern that has different amounts of structure. This would be a clustered part of the pattern, and this would be a hyper-dispersed part of the pattern, and those are separated by occurring at different spatial scales. And so this would be of great interest to us if we could assess things in this way. So just to give perhaps, or this in turn requires new approaches to sampling, point patterns, and we will discuss these more nuanced approaches in our next video. Thanks. See you next time.